Hello everyone, I'm Mayan Asuncion from BSN1 Swanson. For today's video, we will be dissecting a pig's kidney and examine its external and internal structures. So let's start. This is our pig's kidney, and this indented part is the renal hilum. Located in the renal hilum, we have the ureter, which we have a little portion left in this kidney. We also have the renal vein, which connects the kidney to the inferior vena cava. And next is the renal artery, which connects the kidney to the aorta. So to differentiate the two, the renal artery is thicker compared to the renal vein. Okay, so the next thing that we're going to do is to cut open the kidney so that we can examine the internal structures. Alright, so we've already made a frontal cut of our kidney and we've exposed the internal structures. So now we're going to examine them one by one. Inside the kidney, we have the three main regions. This outer region of our kidney is called the renal cortex. Inside our cortex, we have the renal medulla. Next, we have this funnel-shaped region at the center of our kidney, which we call the renal pelvis. Now, going back to the renal cortex, inside our cortex, we have these triangular-shaped structures called the renal pyramids. In between the renal pyramid, we have the renal column, and below it, we have the white portion known as the minor calyx. The larger portion where all the minor calyxes meet is the major calyx. This calyx's main function is to funnel urine towards the entire portion here, which is our renal pelvis. Now we're going to trace the different blood vessels in the kidney. And as we go through, I will show you the path taken by the blood and the filtrate that becomes the urine. The journey begins with the renal artery, which carries oxygenated blood from the aorta into the kidney. This blood is rich in nutrients and contains waste products that must be filtered out. The renal artery divides into segmental arteries, which become the interlobar arteries, running through the renal columns, which is right here. The interlobar arteries become the arcuate arteries, arching along the base of the renal pyramids. The base of the pyramid is right here. The arcuate arteries give rise to the cortical radiate arteries, extending into the cortex. The cortical radiate arteries lead to the afferent arteriole that supplies blood to the nephrons, where blood is filtered by the glomerulus, removing waste products. The filtered blood then exits the glomerulus through the efferent arteriole. Then it enters the peritubular capillaries, which surround the renal tubules. From the peritubular capillaries, the blood proceeds into the cortical radiate veins, which are located right here in the cortex. Combined, it will form the arcuate veins that merge to become the interlobar veins running through the renal columns. The interlobar veins eventually join to form the renal vein, which is connected to the inferior vena cava. The blood is then carried from the inferior vena cava to the lungs, where it is replenished with oxygen. After filtration, the filtrate containing substances and waste is collected in the glomerular capsule and leaves via the renal tubule. The filtrate enters the proximal convoluted tubule, where reabsorption of important substances occurs. The filtrate then moves to the loop of Henle, located in the medulla, which helps regulate water and electrolyte balance. The filtrate continues to the distal convoluted tubule, where more selective reabsorption and secretion occur. The filtrate flows into the collecting duct, where further adjustments to water and solute concentration occur. Urine is eventually collected in the renal pelvis and then transported to the bladder through the ureter. The bladder stores urine until it's ready for micturition or voiding. In this process, the urine exits the body through the urethra. So that's all for our pig's kidney dissection. Thank you very much for watching.